Hello, and welcome to Alexander Disease Research Update, episode number 34, recorded on August 7th, 2026. I'm Albie Messing from the Waisman Center at the University of Wisconsin, and with me today are Jared Cullen and Nadine Connor, both also from Wisconsin, where Jared is a graduate student in the Department of Communication Sciences and Disorders, and Nadine is a professor in that department as well as surgery, and on top of that, has served for many years as the Associate Vice Chancellor for Research Policy and Compliance. Since neither of you have been on the podcast before, can you each say a bit about your backgrounds and how I managed to convince you to work on Alexander disease? Nadine, let's start with you.
Well, I was convinced by Albie to work on Alexander disease because, first of all, I would do pretty much anything for Albie. And second, because we have done some work in the lab on other degenerative types of diseases in the past. And we were really interested to see how Alexander disease would fit into the frameworks that we've been working on in the lab.
And your PhD was in neurophysiology, is that right?
Right. My PhD was in neurophysiology at the University of Wisconsin quite a few years ago. And I began my career as a speech language pathologist, which I still am, a clinically certified speech language pathologist, although
I didn't know that.
Although I haven't practiced clinically for a long time because of my research and administrative responsibilities. And I got involved in speech language pathology sort of on the rebound as a college freshman at the University of Maryland. I was an engineering major, and I was one of two women in my class at that time. And I didn't really connect with engineering, and I took an introductory class in speech and hearing sciences and thought that this is what life is about. It's about communicating with people. It's about being able to eat and swallow. And I felt very passionate about that and got into the neurophys aspect of all of that because I realized just how little was known about brain and muscle mechanisms and how they control these things I was so passionate about, communication and eating and swallowing.
All right. And Jared?
Yeah, so I kind of got involved in this from sort of a non-traditional standpoint. I majored in biology at Edgewood College, and then afterwards worked in several areas of basic science research. I worked as a microbiologist in virology at the Veterinary Diagnostic Lab at Wisconsin. I also worked in cancer genetics before joining Dr. Connor's lab in 2013. And like Nadine said, I also became very interested in how the tongue and head and neck systems support everyday functions of eating, swallowing, and communication. I was particularly fascinated by how complex these behaviors actually are. And that ultimately led me to pursue my PhD in Dr. Connor's lab, where I'm currently studying how neurological disease affects oromotor function and whether targeted rehabilitation can preserve or improve related functions.
Well, thank you both again for being here today. Before we get started, please send feedback and questions to my email, which is amessing@wisc.edu. So today our topic will be a paper on which we're all co-authors, but it was really done in Nadine's lab and with Jared doing most of the work, entitled Behavioral Characterization of Bulbar Sensorimotor Function in a Rat Model of Alexander Disease, which is in press in the journal Behavioral Brain Research. And the key message of this paper for me is, one, that you can do this kind of study at all, and two, that the rats have some of the same problems as humans and in functions that are among the most bothersome to patients, clinically called dysphagia, dysarthria, and dysphonia. So this adds support to using the rat models as a way both to study mechanisms of disease as well as interventions or treatments that can reverse these deficits. Nadine, let's begin with you. And actually, one word in the title perhaps needs a definition. What is meant by bulbar?
Bulbar refers to the bottom part of the brainstem, right on top of the spinal cord. And it was given that name bulbar by early anatomists because it looked like a bulb, simply on the top of the brainstem. So we talk about brainstem mechanisms and bulbar kind of interchangeably now. And that region contains the cranial nerve nuclei that send out nerves that connect to the muscles in the head and neck. They're very important nerves for controlling those muscles.
So the brainstem is one of the most highly conserved structures in the vertebrate nervous system. And so many of these functions really are commonly shared among all vertebrates. Perhaps, Nadine, you could also explain for us, how does oromotor function in rodents? How is that similar to humans? And how have you used those models in the context of your research on Parkinson's?
Yeah, so as Albie just mentioned, the human and the rat anatomy in the brainstem to the muscles to the other structures are highly conserved between humans and rats. That means they're really similar. So we have the same cranial nerve nuclei that I just mentioned, the same nerves that go to the muscles and connect to those muscles in the same way, and they function in the same way in rats and humans. Now, there are some differences that we bear in mind when studying, for instance, swallowing, because as you can imagine, humans, we swallow upright, whereas rats are more horizontal when they swallow. So those geometric differences in the airway are things that we keep in mind when interpreting our data. But we find, and throughout our work in aging, head and neck cancer, Parkinson's disease, and I want to mention the Parkinson's work is with strong collaboration with our colleague Michelle Chucci, who is really the expert in Parkinson's disease. But we have found that a lot of the signs and symptoms, if you will, in the rat recapitulate what has been found clinically and observed clinically in Parkinson's disease. So slowness of movement is one of those things. Changes in eating behaviors is another thing. Changes in muscle structure is another thing. And those things that we find in the rat are similar to the human. So a question I get asked is, well, then why don't we just study this in the human then? Why do we bother working with rats? And the reason for that is because we are looking beyond the clinical and behavioral manifestation of these diseases and how these diseases are treated. We're trying to look at mechanisms. And in order to do that, we need, for instance, to look at the muscle tissues. And we find it difficult to convince humans to give us part of their tongue or, or part of their larynx to study. Also, in rats, we can control a lot of things that can't be controlled in a human study. So we can control age of onset of the disease or disorder. We can even study rats before they manifest signs and symptoms of a disease or a disorder. And you can imagine in Parkinson's disease or other diseases, people don't go to the doctor for their problem before the signs and symptoms manifest. So we're able to do that. We're able to control genetics, whereas you can't do that in a human. We're able to control food intake. We're able to control environmental factors that we can't control in humans, such as family support at home and other things. So the use of rats gives us a really precise experimental control to isolate the variables of interest in our studies.
And I know that both of you have mentioned the implications this has for therapeutic intervention. So that's going to be something everyone's really interested in. We'll come back to that later. Jared, let's start digging into some of the details here. What were the tests that you were able to use with the rats, and what functions are you trying to study?
Yes, so we looked at tongue function and strength. We also looked at mastication, or them doing a pasta biting task. And then we looked at swallowing using video fluoroscopic swallow studies. And then we also looked at vocal output by assessing their ultrasonic vocalizations.
Let's start with tongue function. How exactly do you study tongue function in a rat?
Yeah, I would say that an easy way to picture it is sort of a small strength testing apparatus. We have the rats in the apparatus and they protrude their tongue through an opening and press a small disc that is sensitive to force. When it presses hard enough with the tongue, then a small droplet of water is dispensed onto the disc, and that sort of acts as a water reward, which motivates the rat to press the disc again. So essentially, we're using operant conditioning to teach the rat to press the disc with its tongue. And once the task is learned, we can gradually increase the force required to receive that water reward. The entire time, the apparatus is recording the pressing events, temporal measures, and the force thresholds. So we can quantify tongue force, number of presses, press rate, how performance maybe changes over time, things of that nature.
I should say two things by way of background. In any kind of behavioral studies with rats, and this is somewhat similar to children, you have to deal with motivation. So these rats are mildly thirsty to begin with, and that's why the water is a reward that they want to receive. Also, you just studied adults in this paper. They were all males, about 7 months of age, 10 of the Alexander rats, and 9 of the wild type rats. All right, so that's how you did it. Can you tell us the results?
Yeah, so what we found was compared to wild type controls, the R237H group produced lower maximum voluntary tongue forces and lower average tongue forces per press. They also performed fewer presses overall and had slower press rates. Within the R237H group, press rate declined over time, and that decline was not seen in the wild type controls. So, I mean, overall, we saw reduced tongue strength and reduced sustained performance.
Do you think that implicates endurance or fatigue?
Yeah, I mean, it is consistent with earlier fatigability, but we did not measure fatigue directly. So really, I mean, weakness, coordination, motivation, or some combination of those factors could all be contributing to the overall pattern. So that's future research, I would say.
All right, so the second category of test you did involved mastication, which is a fancy word meaning just chewing and biting. Is that right?
Yeah. So rats naturally bite and chew uncooked vermicelli pasta, and we record the animals eating standardized pieces. We use a microphone, and then we can sync those audio recordings up with video. And from those video recordings, then we can count individual bites and chews and measure how long it takes for the animals to process each piece. It's a fairly straightforward task, but it does give us objective measures of feeding behavior, oral processing.
So this is one of the tests that I was really amazed at when I first saw you do it. So the pasta has to be a certain type. It has to be uncooked. So when they bite, you hear sort of a click. It's a very, very discrete sound. And then The difference between a bite and a chew exactly is what?
So for the purposes of the bite and chew, we did audio recordings, as you said. So we were looking at sort of audio intensity or frequency. And then we were also using the video evidence to inform bites versus chews. So bites were the consuming process. So they would actually hold the pasta piece with both paws and take a bite and consume the pasta piece. And then chewing was, once the bite was taken, the chewing was processing that bite in the oral cavity. So we could differentiate bite and chews based on the audio and then confirm that based on the video.
Okay. Next one is swallowing. How did you do that?
So for swallowing, we essentially record an X-ray video while the rats eat a peanut butter and barium mixture. And the barium is a radio-opaque substance that shows up nicely on the X-ray. And so we can see the portion of food being swallowed and follow its movements. And we can measure a number of different things from that, including oral phase duration and other swallowing kinematics. Things like bolus size, which is basically the size of the portion of the food that's being swallowed, and other kinematics towards the esophagus.
Yeah, and I think this is a test that many people have experienced themselves as well
Sure.
when they're getting worked up for these problems. All right, the last category are vocalizations. I think a lot of people don't realize that rats vocalize unless they actually handle them. And so first, can you explain what types of vocalizations you're able to study and then what you found?
Yeah, so for vocalizations, we are studying social vocalizations in male rats. And to do that, we introduce a receptive female.
You have to explain what a receptive female means.
Okay. Yeah, so we introduce a sexually receptive female, a female that's in estrus that piques the male's interest. Then once we remove the female, it motivates the male to call or vocalize. And this is a social call.
Like, please come back.
Yeah, please come back. Where did you go? That sort of thing. Best one-liners that you could come up with. Insert there. And we have to use a special ultrasonic vocalization microphone because the male rats are calling at frequencies above the range of human hearing. So then we can turn those calls or vocalizations, those recordings, into spectrograms and measure different attributes of the spectrograms, like duration of the vocalization, frequency, intensity, or a number of different acoustic patterns.
So what did you find?
So in the R237H group, the longest vocalizations tended to be shorter, particularly with a simpler call type. They did still produce long calls, they just did so less consistently. That pattern, could be interpreted as reduced vocal endurance or altered coordination. But again, call duration depends on things like respiratory support or control of the larynx. So this study didn't differentiate those actions or which would be primarily responsible.
Did you find anything related to volume or frequency?
No, like total call count, call type, composition, intensity, bandwidth, those we didn't detect differences in any of those.
Maybe we should summarize then what you think is the key message of this paper for everyone. This time, Jared, I'll start with you. What's your key takeaway message?
Well, I think that the model shows a behavioral phenotype. And I think, eating and swallowing and communication may be major concerns for individuals living with Alexander disease. And this model gives us objective measures of function that we can investigate underlying biology or mechanisms, which Dr. Connor's lab is capable of doing, and evaluate interventions aimed at either preserving or improving function, whether that be complementary or not.
And Nadine?
Yeah, I think this paper is a great example of the key questions that we ask in the lab. And those two key questions are, can we understand it? And can we do something about it? So this is the first step in the can we understand it part of our framework. It shows that there are these behavioral differences in Alexander disease affected rodents that do, again, recapitulate a lot of the effects of the disease on the oral motor system that clinicians observe. So it provides a framework for then moving on to question 2, can we do something about it?
Let's touch on question 2 for a moment. Based on your experience with other disorders that you've worked with, aging, Parkinson's, what are the things that you can do to correct these deficits?
There are things that people do clinically that are not well understood from a physiologic basis. And what we try to do in the lab is model interventions, treatments that are used clinically with rodents to see if we can understand the underlying physiology of these treatment effects. What is happening in the muscles, because we really do study things mostly at the muscle level and we work our way back to the brain. We're not really central nervous system people like Albie. We do believe in the biological basis of behavior. That is, the things that you see behaviorally are caused by biology. That's what's doing it. So what are these biological changes that happen with treatment? And if we can identify them, we can optimize them, and we can develop treatments that optimize them, that we can then use as the basis for hypotheses that we test clinically. So we're not saying that what might work in a rat will work in a human, but it does allow us to develop a targeted, focused hypothesis that then is more ethical to test in humans. So that's really our goal. And we do model interventions that are real world clinical interventions, which I can talk about if you want me to, or Jared can.
Yeah, can you just explain what some of those are?
Well, for swallowing disorders or dysphagia, a clinical treatment that's used is tongue exercise. So there are some devices out there that are commercially available. The Iowa Oral Performance Instrument, or the IOPI, is a very popular device that's used clinically. And it allows the user, the human user, to push their tongue against a resistance and to progressively increase that resistance and to get feedback on the device. This has to be prescribed by a speech language pathologist and done in accordance with that speech language pathologist program that they would develop for you and monitor you as you do it. So that is a popular treatment that has shown to have beneficial effects on the swallow in a variety of patient groups. Another treatment that's used clinically is neuromuscular electrical stimulation, where the muscles in the neck and underneath your chin here are stimulated to activate those muscles in the same kind of way that exercising them might do. And that's a treatment that we model in the lab for the tongue. And another treatment that we've also explored in the past is treadmill running. And you might wonder why running on a treadmill would help your swallowing.
I do
And
wonder.
we don't know that it does help your swallowing, but there's good physiologic background to suggest that it might, because what has to happen when you run on a treadmill or otherwise? you have to breathe a lot more. It upregulates the whole airway, including, guess what, the upper airway where the tongue lives. So there is tongue contraction during running that exercises the muscles of the tongue. So in our studies on treadmill, the question was, do you need to directly train the tongue by pushing against a resistance? Or can you simply upregulate the tongue indirectly through treadmill running? So we had a study on that early on. We haven't used the treadmill recently, but that's just an example of the various treatments that we have explored for activating the tongue.
So, I know that many of the Alexander community get speech and language evaluations. I just haven't heard anything from them about the interventions that are being done to improve it. Other than, for instance, consistency of food, types of food and consistency of foods to make it easier to swallow. But in terms of improving the swallowing process, that's what's new to me. So
those are compensatory treatments,
right,
rather than directly addressing the problem. And we would love to move to something that can directly address the issue rather than just compensatory types of changes. And this is something that Jared is hoping to, well, that he not just is hoping to, that he will do in his dissertation with these Alexander rats, is to look at tongue exercise treatments, because we have a way of doing that in the rat.
Well, we look forward to that. That's all for today's episode of Alexander Disease Research Update. Thanks for listening, and thank you to Jarrett and Nadine for joining me today. Our theme song was written by Charlie Allenson, and thanks to Daniel's Fight, our sponsor for these podcasts. I'm Albie Messing. See you next time.